the last speaker, and then we'll have a good 20 minutes for discussion. That's Dr. Babu Valliyodan,
Thank you.
University of Missouri, who will be talking to us about another crop, soybeans.
Good morning, all. Thanks to Rajiv, Anu, and ID5 for me here. We are working with the soybeans, the University of Missouri. We got a National Center for Soybean Biotechnology over there. My team, just following what Darwin and others did long time back, just following the mutations in soybeans, just find out how these mutations vary naturally in different soybean lines. Also in soybeans, the problems are we got the narrow genetic basis is the problem with the soybean. So we have to look at very. Okay, I'm sorry about that. The narrow genetic basis of a soybean is a bottleneck for us to look at the yield or the quality improvement soybean. So the challenge is how we can identify genes or genomic region and help the soybean breeding program. That's our aim. So for that, we developed our own genomic resources. In 2010, we published, the first soybean genome is published. And after that, several soybean genome-related publications or reports available. Then recently, we started a mega project on sequencing a core set of soybean genomes from the United States Soybean Genome Repository. We selected about most diverse soybean lines from the 20,000 accessions what we got. We took about 10% of the, we have a 10% of the core collection we selected for sequencing. So we sequenced the most diverse lines and approximately about 15x genome coverage. Then we compared with the existing reference genome. We found the mapping percentage is close to 99%. Then we have to look at the remaining 1% to see what are the mutations available in soybean. We used we developed a workflow for analyzing soybean genomes, and this is publicly available in SoyKB. Then after sequencing about 600 lines, we found about 13 million SNPs. These SNPs are the key for associating genotype with the phenotype in soybeans. So if when we arranged all the SNPs, we found that soybeans, we can classify soybeans where it is originated from and which group we have to look at. Then we can clearly identify the diversity of soybeans when comparing wild type, landrace, and cultivars. Then we can find all the gene present and absent variations or structural variations in soybeans. So we set the stage in the sense we made a great genomic resource to look at the phenotypes. So back to the biology, as in legumes, it is reported that there are natural genetic variations in root traits. That's true. In soybean also we got a huge genetic variation in root traits. So we developed systems both in greenhouse and field-based systems to look at different root architectural traits. So we do tube-based system in the greenhouses and the shovel-based system in the field. Then we select the specific soybean lines with contrasting root system architectural traits. So this is some of the examples of soybean lines having different root architectural traits we use for genetic dissection. As I mentioned, we developed this 5 million or 7 million SNPs, good quality SNPs from that 600 soybean lines. We use those SNPs matrix to look at associate soybean root system architecture traits and find out what are the genes associated with that. So these are some of the examples I'm giving. So for total root tank, we found some of the genes in chromosome 20 and some on 6. You can see the cutoff value is very stringent. So we find that these are the true genes associated with root length. Same with root branching. We found the new targets. Same with the root angle. We find a very key gene associated with the root angle, and we clone those genes, and the functional characterization is in progress. And also, not only looking at the phenotypic morphological level, we look a little bit deeper into the anatomical level. We screen the soybean lines, what we sequence, as Kurt said, on some of the anatomical changes in soybeans, in the root vasculature. Then we found soybean lines associated with different root-related xylems or vasculature structure. We identified a gene associated with that vasculature, and that associating with the lateral root number. The key is here the soybean lines with the varying vasculature structure have more lateral root number. You can see about more than 3,000 tips you can see in those lines. We went a little bit deeper. We cloned the gene. It's a hormone-related gene, GLR1, and we characterized it in the soybean hairy root transgenic system. We found that yes, this gene contributes root system architecture in soybean. So at present we have about 1,200 soybean lines, the genome sequence available, and we have the SNP matrix available for the 1,200 lines, and that is used for looking at different phenotypes and trait discovery. All this data, soybean phenotypic or genotypic data, is available either SoyKB and in Soybase. So what next? We want to go a little bit more on the soybean genome. Make a haplotype map for soybean genome. Use that haplotype map to dissect the traits like root system architecture or any other drought-related traits, and find out that specific allele and use that allele for further research. So in summary, we have a germplasm set. We developed a genetic variable screening system, and we found out the alleles, and we are using those alleles or markers for the soybean breeding for better yield. And also, we're using those information to develop genome editing platforms. And a couple of the genes what we identified related to root traits, we are trying to edit it, and hopefully we can get a transgenic or modified plant with a better stress tolerance or better root system. This is a huge project. This is our team in the University of Missouri, and we have collaboration with the USDA ARS, and all the genome work we did with BGI and NRGene and BioNano, and we have a collaboration with Rajiv here, Dave in Australia. He's putting together all the 1,200 genomes to get the pan-genomes of a wild and cultivated soybean, and also looking at all the alleles. And we have a collaboration with Brazil. There's another soybean producing country, and we partner with a couple of companies to conduct all the develop all the genomic research. And funding from United Soybean Board and USDA, and all the data is available in KBase, SoyKB, and also iPlant.